So now we are going to talk a bit about proteins, and then we're going to talk about nucleic acids. So let's start with proteins. Remember, we're talking about our biological molecules here. So we've already talked about carbohydrates, and we've talked about lipids, and then we're going to talk about proteins now, and then nucleic acids, and that will be the 4 biological molecules that we're talking about in this section. So for proteins, first we'll talk about the functions of the proteins. And the functions are many, so I'm just going to write many in here, and I'm going to give some examples. But the proteins are the things that are going to be doing most of the things inside of our cells, and therefore most of the things inside of the body. So one of the main functions is for use in metabolism, meaning that our enzymes are mostly proteins. And so remember that proteins, or enzymes rather, are going to be things that are going to speed up a reaction. And it decreases the activation energy, and then it speeds up a reaction in the cell. So whether or not we're talking about building up molecules or breaking down molecules, and then remember that metabolism is all of the chemical reactions in the body. And so then most of our enzymes are made out of protein. So that's a huge an important thing for the body is for metabolism. Support. So structural support. So things like keratin. Keratin is a protein that we find in the skin, on the superficial cells of the skin, so the outer cells of the skin, but also we find large amounts of keratin in our hair and in our nails. That's just one example of a type of protein that's used for structural support. Also transport. So we have proteins in our plasma membranes that make channels. Channels are carrier proteins. And these are going to allow different molecules to move in and out of our cells. So they're useful for transport, for moving things between things. Defense. So we have antibodies. Antibodies are things that go around and they label bad guys in our body and tell our body to destroy them. So proteins or antibodies are proteins. Also for regulation. So regulating parts of the body and controlling parts of the body, and this is because some hormones are proteins. So we did already talk about the sterol hormones, but then some hormones are proteins. Many of them are proteins. And then the last example for motion. We have these proteins that are called actin and then myosin. And those are the major proteins that we find in our muscles that are allowing our muscles to contract and relax, that are allowing us to move, our bodies to move. So those are just kind of some examples of the functions of proteins. So getting back to the structure then, or getting to the structure of proteins. Remember, we have our monomer and polymer. So we talked about the monomers and polymers of carbohydrates, the monomers and polymers of lipids. Now we're going to talk about the monomers of proteins. So the monomer of proteins are amino acids. So we'll take a closer look at the amino acids and what comprises an amino acid and the different types of amino acids. And then when we have our amino acids together, when we talk about our polymer, what we're talking about then is the completed protein. So for proteins, the polymer itself is a protein. And this is our three-dimensional structure. It's very complex. And the complexity and the 3D nature of it is what's going to determine the function. All right, so let's take a look at our monomer, the amino acids, first. All right, so amino acids. So the basic structure of an amino acid is that we have a central carbon with a hydrogen, a carboxyl group, an amino group, remember our functional groups, and an R group, so meaning the rest of the molecule. So let's take a look at some of the amino acids just so we can kind of talk about them. But in general, there are 20 or about 20 amino acids total. The reason that I say about 20 amino acids is there are 20 well-known amino acids, and then there are about 2 or 3 others that they say are pretty well-known, and then there are some that people say are amino acids or not so much. So there are 20 generally recognized amino acids, and they're different amino acids based on their R group. So based on the R group. So the R group, and the reason they're based on the R group is, of course, because all of the amino acids have these other things that we mentioned. So they have the central carbon, they have a hydrogen attached to it, they have the carboxyl group and the amino group, which are all the same depending on, or all the same across all the amino acids. So then the only thing that's going to be different is this R group. So the R groups can be polar. So they can be a polar R group, so have a partial positive or a partial negative charge. They can be nonpolar, so not mixed with water, mixed with fats, or they can be charged. So they can have a positive charge or a negative charge, depending on what's on there and the electrons associated with it. So the R groups determine the structure of a protein. And that is because when we take a look at the structure of the amino acid, so let's take a look at that. So we have our central carbon, then we have a hydrogen coming off of that central carbon, then we have the carboxyl group, this can be the H, or we can just do the, we'll just do the negative to stick with the carboxyl group there. Then we have the amino group. This is typically a positively charged amino group because remember it grabs on that third hydrogen. And then down here at the bottom is where we have the R group. So this entire part up here is what makes it an amino acid, and then this right here is what makes it a specific type of amino acid. So, for example, if we had something like CH3 as the R group, this is actually alanine. That's the amino acid alanine, is all of this stuff, but then the R group is the CH3. Now, this would be an example of a non-polar amino acid. So this would likely interact down here with a lipid portion or a hydrophobic area of a protein. Then let's say, for example, we had something like asparagine, which actually has some more nitrogen on it there. But you can see right here would be a partially negative section. And so this would be a polar group, asparagine. And so this would then interact with water, would be hydrophilic, and would be able to interact with water in that way. Or a more common one, CO minus, another carboxyl group, and this is aspartate. So this would be another example of a polar group that would interact with water when we're talking about a protein. And so we have these different types. We have our nonpolar, we have our polar, and charged groups, which has a negative charge there. And so the reason that the amino acids determine the function of the protein is because when we are going to be combining amino acids, we're going to be putting them together. Remember, the amino acids are the monomer, and we're getting up to the polymer. We're going to take the amino acids and we're going to combine them. We're going to make bonds between the amino acids. So when we do this, when we join one amino acid to another amino acid, what we make is called a polypeptide. So this would actually be our polymer, is a polypeptide. So a polypeptide, which is our string of amino acids held by covalent bonds. And the bond that we're talking about here is specifically called a peptide bond. And that's why this is a polypeptide, poly meaning many, and then peptide bonds, so many peptide bonds. And that's where we are going to be joining by dehydration reactions between the carboxyl of one amino acid and the amino group of the next. So if we take a look at that, we'll have our central carbon, and then we'll put the hydrogen up here for illustrative purposes. Then we have our NH2 or NH3. Then we have the carbon, OH, and then double bonded oxygen. And then we have the same thing over here, carbon, hydrogen, our amino group, our carboxyl group, and then down here is going to be the R group. So if we are going to have a dehydration reaction with these two individual amino acids, then what we end up with down here is, of course, we are going to be using this amino bond and then this carboxyl bond. And when we're talking about dehydration reaction, remember we're talking about losing water in the process. So then what we end up with is we have this amino group, which is going to stay just as it is unless it bonds to another amino acid, which it will or will not depending on where it's at in the chain. And then what we see, so we still have this double bonded oxygen, but then we end up having our peptide bond here with the amino group. And we still have our R groups here. So then this right here, this is our peptide bond. So you can see we've lost part of our carboxyl group, our OH here, and then we've lost one of the hydrogens here. And that's because then we have the water here. So then we've created a peptide bond there. And then, of course, more amino acids are going to be here, and more amino acids are going to be over here, and then we can make a long chain. So polypeptide meaning many peptide bonds. So what we do then is we have enzymes that are going to take these individual amino acids, so one of the 20 or so amino acids, and it's going to attach the next amino acid to it through dehydration synthesis. We're going to lose some water in the process, and then we make a peptide bond. Then it's going to grab onto another amino acid, then it's going to attach it here. So we have another amino acid, then it grabs onto another amino acid and attaches it here. And then the next one, and then the next one, and we end up forming these very, very, very long chains of amino acids. And these very long chains of amino acids are called polypeptides. Now, when we talk about polypeptides, we have already mentioned that the protein shape is going to depend on these interactions. And so if we're talking about the protein shape, we're talking about our 3D shape of the polypeptide. Held by interactions between the R groups. So we have these amino acids that are all going to be, I'm going to shorthand it, so we have the amino acid with the peptide bond, amino acid with the peptide bond, amino acid with the peptide bond, and we have these chains. Now what I'm not showing here is that we have our R groups hanging down. And remember, our R groups can be very different. So let's say this is a polar R group, and then let's say this is a non-polar R group. And then let's say this is another non-polar. And then let's say this is a polar R group. And then let's say this is a positively charged R group. And this is a negatively charged R group. So when we have these different amino acids, it's going to be these interactions between these R groups that are going to start to form the way this protein looks. So what we can have are hydrogen bonds. So hydrogen bonds between polar side chains. Or R groups. We can have ionic bonds between our positive and negatively charged R groups. We can have covalent bonds between our sulfur groups, or sulfur R groups. So if they contain sulfur. So for example, cysteine. We end up making these disulfide bonds with sulfur R groups. It tends to be a strong part of making proteins. Then we also have hydrophobic interactions. And these are going to push these non-polar side chains, or R groups, to the inside. It's going to sequester them and get them away from the water. So remember, inside of our cells, we've mentioned, or in the bloodstream, is mostly made up of water. So if we have, for example, our polar side chain here, let's see, this first one is a polar side chain. Then we can talk about it bending, so this chain might bend so that this one is going to be close to this one, these two polar ones, because polar likes to be near polar. And so we might have some hydrogen bonds between these two polar chains because of a bending of the chain here. And then similarly, these two nonpolar ones are going to try to sequester themselves inside of the protein so they can be far away from the water. And then, of course, these guys are going to have, let's say, an ionic bond between them where they're going to gain and lose electrons, and then they're going to sit right next to each other and stay very close because they have that positive and negative charge. And so if we can imagine taking this chain, and we can go on and on and on with many amino acids this way, many amino acids this way, all being polar or nonpolar, making covalent bonds with the sulfur R groups, the disulfide bonds, hydrophobic interactions where those are trying to get to the inside and not be near the water, hydrogen bonding either with other polar molecules or with water on the outside of the protein. All of these things are going to contribute to the way that the protein looks, the way that it is shaped. And then, as we've talked about with enzymes, it's very, very important for a protein to, or for an enzyme specifically, but for all proteins to have a particular shape. Remember, we had that active site, and if the active site is not the correct shape, like a lock, then the key won't fit in there, so our reactants won't fit in there, and then we can't make our products, and then the protein won't work, or the enzyme won't work. So it's important that we have the correct amino acids in the correct order, and then those interactions will occur to make the correct protein shape. So now I want to go through kind of some general ways that we make the protein. So there are different steps to making a protein that we'll go over briefly. So we start off with our primary structure. So our primary structure is just the sequence of amino acids. So this is our amino acid chain that has the polypeptide bond. So if we're talking about our primary structure, this is the primary or first structure of our protein, is where we take our individual amino acids and we're going to attach them to each other with the peptide bonds. Then what happens next is that we have our secondary structure. And our secondary structure is where we can see some hydrogen bonding between the amino acids. Shorthand it. between the amino acids. And then what we end up getting at the secondary structures, we get either alpha helices or beta pleated sheets. So these are the two different types of structures that we find at the secondary level. So the secondary structure, so 1 primary, 2 secondary. And so what we see are alpha helices or an alpha helix or beta pleated sheets. And what that means is if we take our primary structure, our sequence of amino acids, due to hydrogen bonding, so those polar versus nonpolar, so moving those polar amino acids around to have interactions between them, we end up with either something that looks like a ribbon, which is an alpha helix, or we end up with a pleated sheet, which is going back and forth like this. So we can end up with something that looks like this, which is an alpha helix, or something that goes back and forth and back and forth and back and forth on more of a single plane. So then keep in mind that these are actually chains of amino acids. So these are all the amino acids that we just saw in the primary chain, except they're all in this structure now. And so what we end up seeing is if we have a really, really long chain of amino acids, we can have an alpha helice, then a beta pleated sheet, then another alpha helice, then another beta pleated sheet, then maybe another beta pleated sheet. And so then we have this really long polypeptide that's going to have various amounts of alpha helices and beta pleated sheets, all depending on their side chains, all depending on the R side chains and the hydrogen bonding. So in our secondary structure, we end up with these different types of structures. Then we move on to our tertiary structure. And the tertiary structure is when we're really going to see these interactions with the side chains, so interactions with the different R groups. So we see the covalent bonding between the R groups. We see the different ionic bonding with the R groups. We see other interactions like the nonpolar hydrophobic interactions. And then, of course, we can see still some polar interactions. And so our tertiary structure is when then now what we're going to do is we're taking the alpha helices and the beta pleated sheets and we are folding them on top of themselves. So we have the alpha helice and then we can put a beta pleated sheet is going to bond there, and then an alpha helice and then a beta pleated sheet on there. And so then on our tertiary structure, what we're doing is we're actually creating bonds between the R groups in the alpha helices and the beta pleated sheets based on these things, which are based on the R groups. So up here, this was due to hydrogen bonding. Down here, this would be things between the beta pleated sheets and the alpha helices. So beta pleated sheets, alpha helices. Up here, it's just the hydrogen bonding in the beta pleated sheets itself or in the alpha helices itself. Down here, now we're attaching the alpha helices to the beta pleated sheets through these other types of bonds. Then our fourth level, our quaternary structure. This is where we have two or more folded polypeptides interacting. So we've gotten all the way to the tertiary structure where we have these interactions between the alpha helices and the beta pleated sheets, and now we have this three-dimensional structure at the tertiary structure. Of course, they're all three-dimensional, but this more three-dimensional kind of globular shape. And then what we do is we take multiple of these three-dimensional tertiary structure shapes and put them together. So they can either be several of the same type of polypeptides that are folded, put together, or they can be different ones that are folded and put together. So then what we can end up with are, if these are our tertiary structure, the three-dimensional ones, we can just put several of them together, and then we'll have something like, say, our active site is in the middle here. In this case, we have 4 polypeptides all put together to make a particular molecule, to make a particular protein. Let's say this is an enzyme. A protein enzyme. And so when we're at the quaternary structures, when we would then call it a protein. all the way up until the tertiary structure, at this point we're still calling it a polypeptide. It's just a folded polypeptide at this point. And then it's not until we actually have all of the different groups, all the different polypeptides that are put together to make the end functional protein, which is when we get to the quaternary portion here. So again, just a reminder that protein shape is incredibly important. Shape is important. So anything that changes the shape of a protein may change the function of the protein. So changing the shape may change the function. Now, the only reason that I say may change the function is because sometimes if one amino acid is replaced by a somewhat similar amino acid, then it may be close enough to where the protein will still function. So if we have a polar amino acid and instead we put in a different polar amino acid, then it may still function the same way if it's close enough to that one polar amino acid. So sometimes we have mistakes, we have issues that happen when we make our amino acid chains that then will incorrectly fold the protein, and then the protein would be non-functional. So some ways that the shape may change, as we just mentioned, would be like changing the amino acid. So changing an amino acid. So as we just went through, it's important that in a particular position there's a polar amino acid, or in a particular position there's a nonpolar amino acid, or a positively charged or negatively charged. And all of that stuff is going to determine the shape of the protein. And just like we mentioned with the enzyme specifically, it's like a key and a lock situation. So we need to have that lock be specific to that key. And if it's not, then it will not be a functional protein. So some things that might, some other things that might change the shape of a protein would be something like pH. So if the pH is improper for that particular protein, if the pH is too high or too low, then it can change the shape of the protein. Something like temperature. If a person has a really, really, really high temperature and it stays that way for a long time, we can start to denature proteins. We see this when we cook food that has protein in it. What we're doing is we're breaking down the proteins, denaturing the proteins using temperature. Detergent. There are different types of detergents that are going to start to break the bonds and interact with the bonds within the protein. Different charges. So if we have different charges, if we have an overwhelming amount of positive ions or negative ions in a particular area, it might start to adjust the way that a protein is folded. Things like mechanical stress, so basically just pulling it apart. So if we tear a muscle, for example, we might be tearing the proteins in our muscle just through mechanical stress, or something binding to the protein. So again, we have lots of things that are floating around that are going to be charged, or that are going to be polar or be nonpolar. And so then we have the ability for things to bind to the protein as it's trying to fold up, and then that would make it be a non-functional protein, for example. I do want to mention, as far as changing an amino acid, there's something called sickle cell disease. And in sickle cell disease, or sickle cell anemia, disease or anemia, what we have is a one single amino acid change. So when we're making the polypeptide, the amino acid chain for hemoglobin, then we have one single amino acid that is not placed properly. And then this changes the whole being of the body. So hemoglobin is a protein. It's made up of 4 subunits. So we have hemoglobin here. And so this would be one polypeptide with its beta-pleated sheets, alpha helices, beta-pleated sheets, beta-pleated sheets, alpha helices, beta-pleated sheets. And so then all together we have hemoglobin. Now there's a heme group in here, and in this heme group, then this is where we have an iron molecule, and that's going to attach oxygen to it. And then that's how it travels around the body in blood cells. So this is an example. This is hemoglobin. Now our normal hemoglobin has these 4 subunits, and if everything is okay, we use the correct amino acids, then hemoglobin is made, fills up the red blood cells with hemoglobin, and then we're able to transfer oxygen very efficiently from the lungs to the parts of the body. Now, if we have this one single amino acid change, then instead of having our chain here, if this one were to change, then what ends up happening is that our strands here, the protein strands, end up becoming a different shape. So they end up, the interactions between the molecules of the different hemoglobin subunits end up crystallizing. And then what ends up happening is we end up having these long kind of crystallized chains of hemoglobin. And then it actually ends up having a sickle shape to the blood cell rather than a nice round blood cell, kind of donut-looking shape. And then what happens is these are very stiff and they do not hold a lot of oxygen, so that means that a person is going to have a difficult time getting oxygen to their tissues, difficult time breathing. And then also these sickle cell red blood cells actually end up getting stuck in capillaries and can cut off blood flow to different regions of the body. So it can be a serious issue, and it comes down to a single amino acid change. So again, shape is very important for proteins. All right, so now we are going to move on to our last of our biological molecules group, and that is the nucleic acids. So our nucleic acids, when we're talking about nucleic acids, what we're really talking about are DNA, RNA, and then also we're going to talk a bit about ATP. So those are the 3 types of nucleic acids, or nucleic-like acids, that we'll be discussing. So the main function of nucleic acids in general, let's see if we look at the function. We're talking specifically here about DNA and RNA. ATP is a little bit different, but DNA and RNA is to store information. So storing information about how to make proteins. So our DNA is going to store, it's kind of our main storage form of information for how to make all of the proteins that are inside of a cell. And then our RNA is going to utilize that information in DNA and be a part of the process of making that protein, or making all of these proteins. Now ATP is a little bit different. We're going to talk about ATP toward the end, after we talk about both DNA and RNA. So DNA and RNA is storing information about how to make proteins, and we still have the same type of monomer and polymer situation. So the polymers are the DNA and RNA. Those are the polymer nucleic acids. Now if we talk about the monomers, the monomer of both DNA and RNA are nucleotides. So what is a nucleotide? Let's take a look at the structure of a nucleotide. So a nucleotide, it consists of 3 different parts. So we have a sugar, we have a phosphate group, also we mentioned it as the phosphoryl group, and we have a base. Now what we mean by base is this is a nitrogenous base. What that means is that it's nitrogen containing. So it contains nitrogen and it's the base of that nucleotide. So let's go ahead and draw a nucleotide here. So this one that I'm going to be drawing, the sugar in this case is going to be a ribose sugar. So if you remember, when when we talked about our sugars, our carbohydrates, our monomers, we talked about our glucose, galactose, we talked about fructose, and then we said the last one was a little bit different, and that was ribose. And this is where we're going to see ribose now as part of our nucleic acids. Specifically, we're going to see it in RNA, but we'll talk about the differences between DNA and RNA in just a moment. So for illustrative purposes here, we just are going to use ribose. So here we have our sugar, and of course these have hydrogens on them. So hydrogen here, and then over here we have the hydrogen. So right here, this is our sugar, our ribose sugar. Here we have our nitrogen-containing base. And then connected over here, we have our phosphate group. And of course, this is a carbon here at the point, carbons on the points, the shorthand there. All right, so then we have our phosphate here. So this is the structure here of our monomer of our nucleic acids, which is the nucleotide. So this is our general nucleotide. So it stays the same in all of the monomers are that we have a sugar. That's the sugar here. And we can have either ribose or deoxyribose for the two different types of sugars, and we'll take a look at both of those. We have the phosphate group, which is the same regardless of whether or not it is using ribose or deoxyribose to connect to. Whether or not it's in DNA or RNA, the phosphate group is the same. And then we have our nitrogen-containing base, and that's where we're going to see some differences, just like we saw differences in the R group of the amino acids. So let's take a look at what these nitrogenous bases and what this nucleotide as a whole, how it works. So this again is our monomer, and if you remember back to what we've been talking about with all of our biological molecules, we have our monomers, and then they're going to be connected to each other to make the polymer. So what we see then is that we have a bond that's going to form between the phosphate of one nucleotide, so the phosphate here of one nucleotide, and the sugar of the next nucleotide. So what we end up seeing is that this phosphate here is going to be connected to this part of the sugar and the next nitrogenous base. And so then we have what we would then see, we'll pretend here, would then be this phosphate group, which is then connected to sugar here, which has OH, which is then connected to the next phosphate group, and then our sugar. And so then what we end up having is this long chain, similar to what we saw in our amino acids. Now what we didn't account for here are the bases, and so we then have the bases that are going to be sticking off the side here. Of course, this is a little bit rough on the edge here, and we'll take a closer look at what this really looks like in just a moment. So just to start off with, we're mentioning the bond is going to form between the phosphate of the one nucleotide and the sugar of the next nucleotide. So this ends up having this kind of phosphate, phosphate, sugar, kind of alternating, and then our bases are all going to be facing this direction. What we call this then is this is our sugar phosphate backbone. Our sugar phosphate backbone. I guess that's not a slash here. Let me write this correctly here. Our sugar phosphate backbone here, where we have our alternating phosphate, sugar, and then the nitrogenous bases are sticking out to the side. So let's take a closer look at how this relates specifically to DNA. We'll get into a little bit more detail. So DNA, one of our nucleotides, or rather one of our nucleic acids, stands for deoxyribonucleic acid. So DNA. Deoxyribonucleic acid is our DNA. So in this case, remember our nucleotide is our monomer, which includes the sugar. In this case, our sugar is going to be the deoxyribose. We have the phosphate group. That's the same in both RNA and DNA. And then we have our nitrogenous bases. Now in this case, we already mentioned that our sugar is deoxyribose, and then our bases are actually going to be A, T, C, and G. Now these are shorthand, so let's take a look at what this really means. So we use A as shorthand, and what that stands for is adenine. So one of our nitrogenous bases is adenine. I could draw this out for you. It's a carbon ring with a 5-carbon ring here. There are actually nitrogens on these corners here. There's a nitrogen up here. There are a couple double bonds in here as well. Let's see, double bond. Oh, there's a nitrogen down there. So this would be our adenine, and you don't have to memorize what the different nitrogenous bases look like. I just kind of want to give you an idea of the different bases, and that their structures are different. So we have adenine. This is what our adenine looks like. And so this is going to be attached to our sugar, our deoxyribose in this case. Then we have T, which stands for thymine. Thymine, I could draw really quickly also. Our sugar here, we've got some nitrogens in here as well. Lots of nitrogen in our nitrogenous bases. All right, looks something like that. So that's our thymine. Got some double bonds in here as well, however. There we go. So thymine, adenine. And then we have C, which is cytosine. And cytosine looks like this. Nitrogen down at the bottom. Nitrogen over here. Got amino group up there. A couple of double bonds in here. So we have cytosine. And then lastly, G is guanine. Guanine looks similar to adenine. Nitrogens, 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 amino group. All right, so we have our adenine, our thymine, our cytosine, and our guanine. And the reason that I wrote them in this order is because our A always pairs up with our T. Our C always pairs up with the G. If you'll notice that our adenine and our guanine are very similar in that they are two-ringed structures, and our thymine and our cytosine are very similar in that they're a single-ringed structure. So you can see that we have a single-ringed structure that's going to have hydrogen bonds with, and we'll see this in a moment, with a double-ring structure. And it always matches up C to G. And then up here we have our single-ring structure matching up with, through hydrogen bonds, our double-ring structure, and it's always A and T in DNA. So adenine and thymine always pair up, and cytosine and guanine always pair up. So these are our nitrogenous bases of DNA. And then let's take a closer look at our deoxyribose. So our sugar, our deoxyribose sugar, is going to look something like this. Now we have our hydroxyl groups on here, hydrogen on here. And remember, where there aren't other things, there are hydrogens, right? So there's a hydrogen here, hydrogen here, hydrogen here. There are two hydrogens here. The corners are carbons, etc. So here, this is our deoxyribose sugar. And you can see the difference between the deoxyribose sugar that has been drawn and our ribose sugar that I had previously drawn, but I'll draw again here. So if you look at the difference here, the difference is this guy right here. So if we were talking about deoxyribose, then we've got a hydrogen down here, and if we're talking about our ribose sugar, we have a hydroxyl group down here. So that's our main difference between the deoxyribose sugar and the ribose sugar. So again, since we're talking about DNA, we're going to be utilizing the deoxyribose sugar. That's going to be connected to the phosphate group, and it's also going to be connected to one of these nitrogenous bases. So then when we look at the structure of DNA, we have our nucleotides that we just talked about, which are monomers. And now when we talk about the actual structure of DNA as a whole, again, we have our phosphate sugar backbone, or sugar phosphate backbone. I'm going to simplify these pieces here so it's a little bit easier to take a look at. So we're going to say this is our phosphate group, and then that would be connected to our sugar. And then we have our phosphate group, and our sugar, and a phosphate group, and a sugar. And then, remember that coming off of the sugar on this side is where we have our nitrogenous base. All right, so nitrogenous base. Nitrogenous base. Oops, they are not coming off of the phosphate groups. That is incorrect. Let me back up there. Sorry about that. I got a little ahead of myself. Coming off of the sugars, of course, so let me put the S's on here. Coming off of the sugars, not the phosphate groups, we have our nitrogenous bases. I apologize. There we go. Let's do one more phosphate, one more sugar with one more nitrogenous base. So let's just pretend this is like a C, cytosine, cytosine, and this is like a guanine. Okay. So then when we have our nucleotides that are going to be matched up, so as you can see here, this is where we have the phosphate group is going to attach to the sugar of the nucleotide previous to it, because this is our entire nucleotide here. So there's one nucleotide, and it's going to be attached to the next nucleotide at this point here, between the sugar of one nucleotide and the phosphate group of the other. And then we can continue on. You have the same bond here between the sugar of one and the phosphate group of the other, and then the same thing down here, the sugar of one and the phosphate group of the other. And then again, this here, this portion, is our sugar phosphate backbone. And then we have our nitrogenous bases that are hanging off on this side. Now what we said was that our A always matches up with our T, and our C always matches up with our G. And this is because, in reality, what we have here is DNA is a double-stranded helix. So the actual structure of DNA is that there are two complementary strands. Two complementary strands of DNA that form a double helix. So what we see is that if we were to move this over for just a moment, we can get rid of some of these arrows here. What we would see then is that we have another strand that's on the opposite side. And so we would see that there is a phosphate attached to a sugar, attached to a phosphate, and sugar, and phosphate, and a sugar, and a phosphate, and a sugar. And so then attached to those phosphates and sugars, we have cytosines and guanines, just because I used those two in this example, but we'd also have adenines and thymines as well. And so in this case, we're talking about guanines and cytosines, are actually going to form hydrogen bonds in the inside here. And so now you can see that what we've done is we have our sugar phosphate going on this side of the nucleic acid of the DNA. We have our nitrogenous bases sticking out off the side there. On the other strand, we have our sugar phosphate bone, and we have our nitrogenous bases. And then our nitrogenous bases are going to end up with hydrogen bonds between them. And so then these hydrogen bonds are what are holding the two DNA strands together. So we actually have hydrogen bonds here. It's not covalent bonding or ionic. They're hydrogen bonds, which means that they can break relatively easily, which is important because we want to do this, and we'll see that in cell processes later on in another unit. But we want to go in and we kind of unzip the DNA at some points, and then we're going to make copies of the DNA. But then they zip right back together because of these hydrogen bonds. And then also remember that DNA is a helix. So it has this helical form like we saw in proteins, but it's a double helix. So we have the two strands that are going to be forming the double helix, where they're twisting around within themselves there. So we see our double strand of DNA. It's held together, so we can say that here. They are held together by hydrogen bonds. And these are the hydrogen bonds that are between the bases. So again, our A and T are going to form hydrogen bonds. They actually form two hydrogen bonds. And our C and G are going to form three hydrogen bonds. And that's why I did the three kind of hydrogen bond dots over here. So A and T and C and G. And so they will form across from each other like we just saw over here in this drawing here. Now you only use cytosine and guanine in this example, but of course we would have adenine and thymine that would match up as well. So this then is our polymer. Our DNA is our polymer. It's a double helix structure utilizing our monomers, which are the nucleotides. Now, where is DNA located? It's located in the cell, but it's located in the nucleus of the cell. And we're going to take a closer look at DNA and how it works and what we use it for, as well as where it's located in future units. So for now, our location of DNA is in the nucleus of the cell. And the function of DNA, and we've already talked about the function of DNA and RNA as storing information, but again, so it holds the information, holds or stores information for making all the proteins, only the proteins, needed to make in an organism. So every single protein that an organism needs to live is stored as information in DNA. And it's stored as information based on the arrangement of those nucleic acids, based on the arrangement of the A's, the T's, the C's, and the G's. So it's like a code. For example, a code might be ATGC, CCTTA, and that would code for some aspect of a protein or some type of protein. But then we might have something that's AAACGGGTA, and this would code for a different type of protein. So we have different letters in different arrangements, and letters, by letters, of course, I mean nitrogenous bases, in different arrangements are going to provide for the different type of protein structures. They actually code for the amino acids in our proteins. So just a little bit more information about DNA is that we have DNA is stored in chromosomes. So a chromosome is a long piece of DNA that contains several genes. So our chromosomes are long, long pieces of DNA, and they contain several genes. So what's a gene? A gene is a section of DNA that contains the information for a single protein. A section of DNA that contains the information for one protein. So we have these long strands of DNA, right? So that would be our backbone, and then let's say we have the different bases here. I don't know what let's say red is A, yellow is T, so it's going to be across from the A's. A's and T's are matched up. And then we have C's. I don't know why they're all on the one side here. I guess because I'm not very creative, but A's and T's and C's and G's. So we have this long DNA strand that consists of all of our A's and T's and C's and G's. And then we can take this, let's say this is a very, very, very long strand, and let's say this is our entire chromosome. Now we have 23, humans have 23 chromosomes. So 23 different chromosomes, and that equals about 30,000 different genes. So if we pretend that this is a single chromosome, so from here to here is one of our chromosomes, one chunk of DNA, one long piece of DNA, then we would say that, let's say if we had from here to here would be a gene. And remember, a gene is a section of this DNA that's going to code for a particular protein. So we would say that from here to here, these nucleotides, whatever code this nucleotide is going to code for a particular protein. So let's say a protein in the skin or something. So this would be our keratin protein, let's say. Oops. And then there would be another gene. Let's say there's a gene from here to here. So there's another gene here on this chromosome. And then there's another gene from here to here on the chromosome. And so then this chromosome would say, let's say there's another one from here to here. Let's say we have 4 different genes on this chromosome, and each of these different genes is going to code for a different protein that's necessary for life in the human body. And then again, we have 23 different chromosomes, and we have 30,000 different genes. So obviously my illustration here is greatly scaled down for illustrative purposes. Our chromosomes are very, very, very long, containing thousands and thousands of bases that code for many, many, many genes. All right, so now that we've taken a look at DNA, our double-stranded DNA, and we know that our monomer is the nucleotide, and we remember it has the phosphate sugar backbone with our nitrogenous base. Now we're going to take a look at the second nucleic acid, which is RNA. So RNA stands for ribonucleic acid. So remember, DNA stood for deoxyribonucleic acid. Deoxy meaning that's the type of sugar we're using. And then ribo, so it's deoxyribose, and it's a nucleic acid. In this case, RNA is a ribonucleic acid. So remember that in RNA, we use the ribose sugar. So the sugar we use here is ribose, as the name implies. And then we have our nitrogenous bases as well in our RNA. In this case, we have our cytosine, which was our C, and our guanine, which is our G. We have our adenine, which is our A. And instead of thymine, which is what we saw in DNA, in RNA we have something called uracil, U. So here is one major difference between RNA and DNA. We already know the first difference between RNA and DNA is the sugar, right? In DNA we have deoxyribose. The second large difference between RNA and DNA is that instead of thymine, which was our T, we have uracil. So uracil in RNA. All right. So then the structure of it is the same. We also have our phosphate group. And our phosphate group is attached to the sugar, and the sugar is attached to our base. And in this case, we have C and G, A and U. Now the structure of RNA is a little bit different than DNA. So the structure of RNA is a single strand. So it's one strand that hydrogen bonds to itself and then can form these complex different shapes. So it's one strand instead of a double strand. So one strand, and it hydrogen bonds to itself. form complex shapes. And similar to lots of other things we've looked at, namely proteins, different shapes are going to allow for different functions. All right, so if we take a look at RNA, then what we have is going to be like our phosphate group, sugar group, phosphate group, sugar group, phosphate group, sugar group, and then as we've seen before, we have, let's see, cytosine, whoops, not on the phosphate, take that back, I messed that up again. On the sugar, right, we have our cytosine, let's say here, we have our guanine, then let's say here, just for fun, we have our uracil. I like to make uracil yellow. All right. So in this case, we stay single-stranded then. We don't have any hydrogen bonding to a double strand of RNA. There is no double strand of RNA. We have a single strand of RNA, but then what we see is that these things can fold up on themselves. So then here, this guy is going to maybe curl around, and it will form those same types of hydrogen bonds between the cytosine and the guanine that you would see in the double strand of DNA between cytosine and guanine. But in this case, it's a single strand, and it's going to fold up on itself, and it will form different types of shapes. And we'll take a look at some of the different types of shapes when we get in a little bit further into the semester. But for now, in just a moment, we'll talk about the different types of RNA. It's important that you know that they can fold up and they'll form different shapes, and then those different shapes are going to give them different types of abilities, different types of functions. So the location of RNA, remember the location of DNA was in the nucleus of the cell. The location of RNA is that it is actually made in the nucleus, but then it does its functions. It functions in the cytoplasm. So we'll take a look at cells a little bit closer in the unit, cells. But we have our outer plasma membrane that we've already talked about when we talked about lipids. And then in the inside we have our nucleus. So this would be nucleus. And in the nucleus, this is where we have our DNA. Now our RNA is going to be made in the nucleus, so it's going to be made in the nucleus in here, and then it leaves the nucleus and it goes into the cytoplasm. And the cytoplasm is in this outer region of the cell. So outside of the nucleus is where we have the cytoplasm, and this is where we have all of the other organelles in the cell that we'll talk about, and that's why RNA leaves the nucleus and moves into the cytoplasm so it can interact with other parts of the cell. So the function of RNA, so once it gets into the cytoplasm, is that they are temporary copies of short sections of DNA used to make a protein. So what we do is that we take DNA, we have our double-stranded DNA. We take DNA and we make kind of a copy of it. So we kind of jot down the information in DNA. When we jot down the information in DNA, what we're doing is we're making a single strand of RNA. That RNA will then leave the nucleus and go into the cytoplasm. And depending on the type of RNA it is, so the copy of DNA that we have, is going to determine what specific function it has in the cell. And there are 3 different types of RNA that we are going to talk about. So the first type of RNA is called messenger RNA. Oops. So we do a little m, messenger RNA is the short name for that. So our first one, messenger RNA. Now messenger RNA, or mRNA, is the one that's going to contain the information about the amino acids in the protein. So this contains information about the amino acid in a protein. So messenger RNA, as its name implies, is actually just a messenger. So it takes the information that's stored in DNA, it copies it down, and it's going to act as a messenger. It's going to leave the nucleus and bring that information into the cytoplasm. And the information that it's bringing is what kinds of amino acids we need to put together in a chain to make a particular protein. Now, the second type of RNA is called transfer RNA, or tRNA. Now, tRNA is going to bind to the specific amino acid and match them up with the information that we have in the mRNA. So it's kind of transferring the information. It's going to grab onto the amino acid and bring it to the RNA to match it up. So it binds to a specific amino acid and matches them to the mRNA. And we're going to take a closer look at how all of this works when we get to cell processes and making proteins. So we'll elaborate on this. But for now, you should understand the differences between the messenger RNA, the transfer RNA, and then the last one, which is ribosomal RNA. This is rRNA. So our rRNA is actually part of the ribosome, which is a different type of organelle, not really an organelle, but lumped together with organelles, which is a type of machinery that's inside of a cell. Now, the ribosome is what's going to make the bonds between the amino acids as part of our polypeptide chain. So our ribosomal RNA, or our rRNA, is part of the ribosome, and it's going to catalyze the reaction that joins the amino acids. So part of ribosome and catalyzes the reaction joining amino acids. So those are our three types of RNA. So again, they are single-stranded. They are going to hydrogen bond to themselves, making different shapes, and that's why we see the different shapes between like the ribosomal RNA, the transfer RNA. Messenger RNA tends to kind of stay single-stranded without bending up too much, but can bend up a bit depending on what we're trying to get to. So that is our RNA section. So we've talked about DNA, we've talked about RNA, and now what we're going to talk about is the last kind of nucleic acid, which is ATP. So ATP stands for adenosine triphosphate. So ATP, adenosine triphosphate. So the structure of adenosine triphosphate is that we have an adenine base. So adenine, just like our other bases, the nitrogenous bases that we saw in both DNA and RNA. So we have an adenine base. It has a ribose sugar. So not the deoxyribose, but specifically the ribose sugar. And then it has 3 phosphates. And that's why we have the tri here, 3 phosphates. So adenosine is related to the adenine, and then our 3 phosphates. So if we take a look at what that looks like, we'll do a quick drawing here of our ribose sugar. And we're just going to kind of put, this is our adenine here, coming off of the sugar. And then over here, this is where we have our 3 phosphate groups. All right. So you can see this is 1 phosphate group, another phosphate group, and the 3rd phosphate group. Right here, this is our ribose sugar, and then here is our adenine, or nitrogenous base in this case. So here is our adenosine triphosphate. So before we get into how the adenosine triphosphate works, or what it is, or how it is that it does what it does, let's talk about what it does, so the function. So the function of ATP is that it provides energy. So it provides energy for almost every process in the cell that requires energy. So all of these things that are in the cell that require energy, this is what we use. This is our energy storage molecule. So it provides energy for cell processes. So when we talk about this, it's needed for things like transporting things. It's needed for our anabolic reactions. It's needed for muscle contraction, so heart muscle, as well as skeletal muscle, and lots and lots and lots of things. And we're going to get into some of the specifics as we move throughout the semester. So ATP is considered our high energy molecule. And the reason that it's a high energy molecule is because of these three phosphates here. The way that this works is that these three phosphates, if you look at them, perhaps you'll notice that there are a lot of negatively charged items here. Negatively charged, and then over here we've even got partial negative charges, lots of negative oxygens on our phosphate groups. Now, if we think of magnets, there's a positive charge and a negative charge. If we take two magnets and we put them together, the positive is attracted to the negative, just like we saw in our ionic bonds, for example, or even our hydrogen bonds with a partial positive, partial negative. Now, what happens when you take a magnet and you flip it around to the other side and you try to then touch it to the other magnet? So you have two magnets, one of them you flip around, and you try to put them next to each other. Well, they repel each other. A lot of times if you have strong magnets, you can't even get them very close to each other. You can push and push and push as hard as you can with your hands, with your arms, and you can't get them close to each other. And that's because they're repelling each other, because negative sides don't want to be next to negative sides, and positive sides don't want to be next to positive sides. They repel each other. They push each other away. Positive wants to be by negative and vice versa. And so what we see here in this ATP molecule is that we have a whole lot of negative charge, a lot of negative charge, a lot of negative charge. And what we've done in the process of making ATP, in the process of connecting these phosphates together and onto our ribose, we have forced them to have negative next to negative. So it's like if we took those two magnets and we pushed them as close as we could together, and we took a bunch of duct tape and wrapped it around and wrapped it around and wrapped it around to where they couldn't pull apart. Now, what we would know there is that perhaps if we wrap them around a certain way, say we did it with some string or something, and then we took scissors and we just clipped the string, those magnets would fly apart. And that's exactly what we see with ATP. We forced these three negative groups next to each other, and they really don't want to be there. So it's like loading up a spring. If we had this big spring and we forced it together with our hands, we've pushed them to be next to each other. Then what ends up happening is we have put all of this potential energy in there to where if we just release our hands, the spring will go flying. Or if we just clip that string, the magnets go flying. And that's because there's a whole lot of energy that's released in a big burst. And that's what we have in ATP. It's a high energy molecule because we have three bonds here, three phosphate groups, that as we break or as we force these into bonds here, there's a whole lot of energy in there. And then once we go in and we remove a phosphate group, what we end up doing is we end up cutting off a phosphate group that releases a lot of energy. So when we remove the end phosphate group, it releases a lot of energy, because it's been stored up in there as potential energy. So then the products, so after we have gotten rid of, first of all, we get rid of this third phosphate group. When we get rid of the third phosphate group, we've now decreased the energy of the molecule, right? We've gotten rid of some of the energy. There's not quite as much energy being forced in this situation. So we decrease the energy. So we have decreased energy products. And our decreased energy products are low energy ADP. This is adenosine diphosphate. Adenosine diphosphate, di meaning 2, because now we only have these 2 phosphate groups. So we have adenosine diphosphate, and we have a phosphate. Now the cells are going to use the energy from glucose. We're going to, we eat food, we break it down, we get glucose, and in the cell we utilize that glucose to add the phosphate back onto ADP to make it ATP. So we have our ATP, which is our high energy molecule. When we get rid of the ATP, or that last phosphate, we break it off. We end up with ADP and the phosphate group. And then when we use ATP, or when we eat glucose, we have glucose in the bloodstream, our cells use glucose to go back to ATP then. And then that ATP is going to be our high energy molecule again, that we can use for whatever we need to use it for. So for, say, building up something, anabolic reactions, right? Building up something that needs energy. So if we take a look at that, let's say I'm going to do the shorthand drawing now. So we have our ribose, we have our adenine, and we have our phosphate, phosphate, phosphate. So we have our ATP here. So this is our ATP. If we go in and we cleave this third phosphate off, we're going to release all that energy, just like those 2 magnets. So if we go in and we break off that phosphate group, we're going to release energy. Then what we end up with, ribose. We still have the ribose, we still have the adenine. And then now we have 2 phosphates, right? So this is our ADP plus our phosphate group. And then what we do is we utilize glucose, breaking down the glucose, we use the energy that we get from glucose to then reconnect this phosphate group. And that requires energy. So then we're recycling here. We have our ATP that we can use when we cleave off that phosphate group. This is going to release a burst of energy that we can use to build up molecules, to transport things for muscle contraction, different things like that. We can utilize that energy, but then we need to replenish that. And we do that by taking in glucose. We use the energy that's in glucose, and then we use that energy to attach that third phosphate again, and that's storing up this high energy molecule again. So this is high energy, cleave off that phosphate group, releases that energy to be used elsewhere. Then we have our low energy phosphate, or ADP, and then once we get glucose, we can put energy back into the system and then make our ATP again. All right, so this is our third and our final piece to our nucleic acids. Just to kind of summarize our nucleic acids, our monomer is the nucleotide, our polymers are DNA and RNA, and we have ATP, which is slightly different. Our DNA is deoxyribonucleic acid, which has the deoxyribose sugar, the phosphate, and the bases. Remember the differences between DNA and RNA. DNA is a double-stranded helix. They're held together by hydrogen bonds and the nitrogenous bases. Our sugar for DNA is deoxyribose. Our bases for DNA are adenine, thymine, cytosine, and guanine, so A, T, C, and G, versus RNA, which is a single strand, does have hydrogen bonding, but to itself. The sugar is ribose. The bases are cytosine, guanine, adenine, and uracil, so C, G, A, and U. And then lastly, we talked about ATP, and that is adenosine triphosphate. So we have our adenine as the nitrogenous base there. It's the only one. We have a ribose sugar, and instead of 1 phosphate, we have 3 phosphates. And then we go between our ATP and our ADP. We release energy, utilizing that energy in the ATP, giving us ADP, and then going back to ATP. So that summarizes our nucleic acids. It also summarizes our proteins. So we talked about proteins, does many, many things, or do many, many things in the body. We have our amino acids, our monomer. We have our polymer, which is our polypeptide, and our final protein. We talked about the structure of amino acids. We talked about our polypeptide and how we make that peptide bond. Then we talked about our different steps to making a final protein, so the primary structure, secondary structure, tertiary, and quaternary structure. And we talked about how important shape is to proteins, and even a single change in amino acid or a change in pH or temperature or anything can affect the shape and the function of the protein.